Welcome to our next set of videos, and the topic here is going to be cell signaling, and some people will describe this as cell communication. So let's get started with this topic then. Okay, so cell signaling or cell communication is all about responsiveness. This is one of the characteristics of all living things. All living things will respond to their environment. That means they've got to receive information from their environment and respond appropriately. Now, when we say environment, that can be the outside world, as we think about the outside of our bodies, or it can be within a group of cells where one cell tells another what an appropriate response might be given a set of conditions in a tissue and an organ. We're going to see cell communication working from the lowliest bacteria all the way up through increasing levels of complexity through simple animals into complex animals, the metazoans, and into plants as well. So across all kingdoms of life, we're going to see organisms responding to their environment. The problem with this topic is the absolutely vast diversity of mechanisms by which cells can send information and other cells can receive that information and act upon it. And so we're going to try and make some generalizations, and we're going to start off with a pretty simple example looking at these two yeast cells. So we'll start off with a simple example, one cell growing in a culture of cells communicating or responding to an environment. So let's consider the yeast cell on the left here. So this is a yeast cell, and there are some molecules which we need to look at. So right here is a molecule called a glucose receptor, and the glucose receptor is a cell surface molecule. So you can ask yourself, how did this molecule get to the cell surface? It's a cell surface protein, and it will bind to glucose. So it has a piece of its structure which sticks out across the cell membrane, across the cell wall, and that piece of the protein will bind to glucose. So if there's any glucose in the environment of this yeast, and yeast love to break down glucose, and we like that too because it makes alcohol. If there's glucose in the environment, it can bind to that receptor. So a receptor is a molecule which can bind to another molecule, usually a small molecule, and we're going to call that a ligand. So a ligand binds to a receptor. So it could be a ligand. Here's a receptor. One binds to the other. So we've got a glucose receptor, and then down here we've got a glucose transporter, and what that will do is it will move glucose. It's one of these transport proteins we looked at earlier, and that will move glucose into the cell. As long as there's a concentration gradient, it'll pump glucose, it'll move glucose into the cell. And then this little blue structure here is the protein that is a metabolic enzyme, and so that's going to be involved in, for example, glycolysis. It may be involved in the metabolism of glucose in the early stages of glycolysis during cellular respiration or as a precursor to fermentation. So those are the important molecules. The last one here is outside the cell. These little purple balls, those are glucose molecules. And so if we've got a yeast cell and it's not got much glucose, then it doesn't make sense for the yeast cell to be making proteins to move glucose into the cell, or to be making proteins which break the glucose down and extract the free energy from the glucose to make ATP. So on the left here, there aren't many metabolic enzymes, there aren't many glucose transporters. Now, if we put in some glucose into the growth environment, into the culture, these yeast grow quite happily just in a little shaker and some media. If we add glucose to that, just powdered glucose, just a couple of millimolar, so you don't have to add a whole lot, a couple of things happen. What we'll see is that the glucose will start binding to these receptors. So the concentration of glucose is now much, much higher than before. Before there was none. And all of these receptors will start binding to the glucose. Now the glucose can bind, and then it will let go, and it will bind, and it will let go. So there's this constant kind of on-off thing. But once the glucose binds to the receptors here, the yeast is going to have some kind of response. And so now the yeast, if the yeast was a human, it'd be saying something like, oh wow, there's all this glucose, and so that means I can now break the glucose down, but hang on a minute, I can't get the glucose into my cytoplasm, so I need some glucose transporters. So these glucose transporters will begin getting expressed in the cell. So the cell will start turning on the genes which make these glucose transporters, and what that means is that all this glucose can now start getting moved into the cytoplasm. So now we've got all these glucose transporters that get expressed in response to the glucose in the growth media. But then the problem for the cell is, well, I don't have any metabolic enzymes to break down the glucose, so now I've got to turn on my genes for these metabolic enzymes, and now the metabolic enzymes start to get made in the cell. And so just in response to adding that glucose, is what we've seen is that this cell is now competent to use the glucose that's in the environment. It can be brought into the cytoplasm, and then it can be broken down by some metabolic enzymes. So cells only respond to their environments in an appropriate way. Well, they should do anyway, is what I should say. So when cells respond in an appropriate way, we have normal homeostatic processes. Now, if cells stop responding in an appropriate way, we start to move towards a disease state. For those of you interested in medicine, pretty much any disease or disorder that you look at is perturbation of homeostasis. Cancer will be a classic example if you'd like to join me and take my cancer biology classes. What we look at are lots of signaling pathways that get messed up, leading to homeostatic imbalance. So that's a real simple example there. The cell's receiving some information, and it responds to the information. So let's ask a quick question. If the cell was responding to the information, what was the information containing molecule in that example? Okay, I'll leave that to answer that one. Let's move on really quickly. Before we get into much more complexity when we look at some signaling in animal cells, I just want to briefly touch on some cell-to-cell communication that you find in plants. This was actually something that Darwin was involved in the discovery of. Here's a nice simple example. You're probably familiar with the fact that plants grow towards the light. In fact, what they're actually doing is growing away from the shade. So if we've got a source of illumination up here, it could be a lamp, in this case it's the sun, then the light rays are striking this side of all of these seedlings. So the light's striking like this, and they then grow towards the light. And so this one here starts off growing straight, but then will bend towards the light, actually bending away from the shade. And so obviously that is going to maximize photosynthetic potential and maximize the number of sugars. So how does that happen? Okay, so Darwin and some others actually found that in the shoot of the seedling, there's a molecule produced, and that molecule we now call auxin, and that molecule diffuses down from the source where it's made, down through the seedling. And what the light does is it either causes the auxin to redistribute on the shaded side, so on this side of the seedling, or it causes a destruction of the auxin on this side of the seedling. Either way, wherever you get an accumulation of auxin, that causes the cells on that side to elongate. And so if you elongate one side of something, you get bending. This is still cell communication. There's a signal being produced up here, information is being produced up here, and cells down here are responding appropriately. So those are a couple of examples to introduce you to some important principles here. The first thing is that cells must respond appropriately to their environments. The environment can be external, or it can be within a tissue, for example. And then we have an information-containing molecule called a ligand, and we have a receptor that receives the information, and then we have a cellular response. So next time, we'll start getting into some more details of this cell signaling. I will speak to you soon.